good weekend, and maybe a little break from biochemistry. I hope to have the exams Wednesday. If not Wednesday, certainly Friday. The answers are online if you care to look at them. And there's another quiz due on Friday. I got a request to put the quizzes on earlier. Apparently, at least one person wants to take the quiz before the class on that material, which is okay. So there'll be a quiz for Friday online this afternoon already. I don't recommend that you take it until after Wednesday's class, but if you want to take it early, you can. Remember, you can only take it once. Right? Once you go in there, that's it. Okay. So today, we're going to get into some more aspects of carbon metabolism. And in particular, we're going to be talking about this pathway called pentose phosphate. And then after that, we'll see how much time we have to get into the pyruvate dehydrogenase complex, which is what happens to pyruvate after glycolysis. But this pathway is one where we have, start again with glucose, and this is glycolysis step 1, and this is glycolysis step 2, etc. Right? So this is phosphohexosisomerase that catalyzes this step. Okay? And so there's another pathway to oxidize glucose, not just glycolysis. Okay? And the other pathway branches off from glucose 6-phosphate to form a compound called, abbreviated 6-PG, but it's called 6-phosphogluconate. And then this goes on to make 5 carbon sugars and NADPH. Okay? Actually, that should be here. So this pathway here. So this is obviously glycolysis over here. This is pentose phosphate over here, and they're branching off this common intermediate, which is glucose 6-phosphate. Okay. And so we could ask, why we're doing this. Why does the cell have 2 pathways to oxidize glucose? This is the pentose phosphate pathway here. It's another way to oxidize glucose. It's particularly active in cells that are dividing a lot and that need to do a lot of anabolic metabolism. That is to say, they need to synthesize a lot of stuff, especially fatty acids, other complex metabolites. And the reducing agent to make those metabolites, that is to say, those metabolites like fatty acids are highly reduced fuels, right? We can burn them, and we'll see how they get burned in pathways that are analogous to the TCA cycle that we'll talk about soon. But if we're going to make them, we need to provide reducing power for them, and the reducing power to make fatty acids is in the form of NADPH. Okay, and so NADPH is made by this pathway here. Pentose phosphate makes NADPH, and so that's one reason why we want it. Glycolysis does not make NADPH. Right? Glycolysis makes NADH without the P, and that NADH is used to, the electrons from that are transferred. If it's anaerobic, they're transferred to pyruvate to make lactate. If it's aerobic, to get transferred down the electron transport chain to make other stuff, to eventually make ATP to get transferred through a series of electron acceptors and donors. But NADPH doesn't have that role. NADPH is used instead by enzymes that build up other complex molecules like fatty acids. So that's one thing we need NADPH for, reductive biosynthesis, meaning biosynthetic reaction that is reductive, that is making something else reduced, that is to say the fatty acid. It also is important because it helps prevent oxidative damage to proteins. These are a couple of examples: red blood cells carrying a lot of oxygen, the eye lens, the cornea, in contact with a lot of oxygen. But oxygen is actually present in the mitochondria because it's the terminal electron acceptor in the electron transport chain that I've been mentioning. So aerobic metabolism, you're breathing oxygen, so it's kind of in all the cells. And when you have oxygen, you also have oxidative damage from the oxygen. And so we need to control that, and there are mechanisms in the cell to control that. I'll show you that in a moment. Another aspect of this pathway, we'll note here, 3 aspects at least more. One is that CO2 is released. That represents the complete oxidation of one of the glucose carbons, right? Because CO2 is as oxidized as you get. So we didn't see that in glycolysis, right? We went to pyruvate, it got oxidized a little bit. We got like 5% of the glucose energy out, and that was it. This pathway is generating 1 CO2, so it's oxidizing all the way. It's also generating this compound, ribose 5-phosphate, which is the sugar in nucleic acids. So one thing that rapidly dividing cells want to do is make a lot of NADPH so that they can make a lot of fats and make a lot of big molecules so that the daughter cells will have those big molecules. And another thing that they need to do is generate the precursors for replicating their DNA, right, so that they can reproduce that way. So this pathway also generates that. Okay. And then finally, it doesn't always generate this. Sometimes it only needs to generate NADPH. Sometimes the cell needs NADPH, but not ribose phosphates. So there's another part of this whole pathway, which is called the non-oxidative phase, which is basically a recycling phase. And that involves enzymes, at least one of which you already know about, an aldolase. Remember, glycolysis has the aldolase that splits the 6-carbon sugar. We've got aldolase again here. And then we've got another enzyme called ketolase, which we'll talk about. So that's kind of the big picture of the pentose phosphate pathway. Gives you some sense of why cells are differentiated to retain such a pathway, and some of the distinctions between this pathway and the glycolysis pathway, even though they're both starting from the same precursors, right? So let's look at the oxidative damage part of things. So O2 is, as I've mentioned before, toxic, and this could happen by way of taking various kinds of different drugs administered for various diseases, radiation. It can happen sort of by accident in the respiration pathway in the mitochondria. But what will happen is free electrons that happen to be around, maybe they shouldn't be, but they are, generate this compound here, which is called the superoxide radical. Okay, and the superoxide radical can be a problem because what happens is you can add protons. So let's say we add 2 protons and another electron, and we'll generate H2O2, which is hydrogen peroxide. And that's quite common, and it's also quite toxic. Okay, so it's kind of the intermediate from what happens when oxygen scoops up a free electron to generate superoxide. So then we have hydrogen peroxide, and at least 3 things can happen to hydrogen peroxide. One of which is the generation of hydroxide radical. This is not enzymatic, right? But it happens if because this is highly reactive. there's electrons and protons around water.